Hello guys, this is Dr. Mobeen. We are continuing with the renal fundamentals. So this is the lecture number 2 in this series about the renal fundamentals, and if you remember last time when we were talking about it, we had gone up to the hemodynamics. So today in this lecture we will talk a little bit more about how the hemodynamics of the renal system are working. So again, if in the kidney, if I asked you to understand one thing clearly, that one thing I would say is the hemodynamics. There are many, the nephron and the pumps in the nephron are very important, the hormonal actions on the kidney are very important, kidneys hormonal functions are very important, the concentration of the urine is very important, the counter current exchange and multipliers are very important, but hemodynamics are the soul of the urinary system. This is what would enable you to understand inulin clearance and creatinine clearance and para-aminohippuric acid clearance and the glucose clearance and the amino acids and other things. So this is a very important concept which we need to talk about. So let's start talking about it without wasting any time. Hemodynamics, so the first thing, so I'm going to erase the nephron here, and I can reuse this diagram over here, where what you're seeing is that this is the heart and the artery is coming out of it, the artery goes into the kidney, and over there, the very first point over there, nephrons are connected in parallel. What that means is that from the heart, when the blood artery reaches the kidney, so let's say this is the kidney, the arterial system here, the renal artery, as it enters the kidney, it starts dividing into, of course you know that it divides into the renal architecture going up to the arcuate veins, arcuate arteries, and from there finally smaller capillaries appear. The important thing to note is that these capillaries are connected in parallel. So this is glomerular number 1, this is glomerulus number 2, this is another glomerulus, this is another glomerulus, this is another, and so on. How many in one kidney? Of course about 1 million. 1 million glomeruli per kidney, and these glomeruli are attached to the artery in parallel. Now remember this, so this is where the confusion is. This is not really very important at this time. What is important is that if I look at one glomerulus, that means a glomerulus or the blood system attached to a nephron, which is showing here as well. So in that case, if I make a fake nephron here like this, then what I'll say is that one glomerulus has afferent arteriole, efferent arteriole, and then peritubular capillaries, and of course then it would become continuous into the vein. So now this is a capillary structure. Remember, glomerulus itself is nothing but capillaries, right? So a small afferent arteriole comes in, divides into multiple capillaries. These capillaries then reunite and they make the efferent arteriole. Efferent arteriole once again breaks up into capillaries, which are called the peritubular capillaries. These capillaries once again then merge back to make the venous system. So this is glomerulus, this is peritubular capillaries, this is afferent arteriole, and this is efferent arteriole. But in here what is important is afferent to glomerulus to efferent to peritubular capillaries. This is a series system. What does that mean? These things are connected in series. These things are connected one after the other. Afferent arteriole comes first, then comes glomerulus, then efferent arteriole, then peritubular capillaries. So going back here, This is a very important point. You need to write it down somewhere and remember it. You should check it off when you have reviewed it. Nephrons are connected in parallel. Each nephron has two arterioles, afferent arteriole, efferent arteriole, and two capillary beds. Glomerulus itself is a capillary bed, and then the peritubular capillaries are capillary bed. So another important thing, two arterioles, two capillary beds. Flow is always equal in series system. So now before this point you should also write it down that a blood supply around one nephron is actually in a series system. Now the characteristics of a series system are very important, and I think we talked about them last time as well. This is such an important topic, the characteristics of a series system. So let's say we have a pipe here. On this pipe you got a couple of faucets attached to it. Let's say this is the input side, and let's say this is the output side. Cool. So this is the structure which we are going to talk about. For your reference, you can say this is afferent arteriole. This is afferent arteriole. This is the musculature of the afferent arteriole, which can change the diameter of afferent arteriole. This is glomerulus, and this is efferent arteriole. This is efferent arteriole, and again this is the musculature of the efferent arteriole, which would control the diameter of the efferent arteriole. So efferent. And then of course peritubular capillaries, which will then become the vein. This is the structure. In this structure, glomerulus is the one that is responsible to allow blood to be filtered. Blood will be filtered. This is capillary. So you know that all capillaries they are fenestrated, they allow the blood to be filtered out of them, and they actually get reabsorption as well. If you look at the systemic capillaries, they have two ends, the arterial end and the venous end. Arterial end allows the blood, oxygen and the other substances, nutrients to be coming out of the capillary. On the venous end, the waste products and the carbon dioxide related things, they are pulled back into the capillaries. So capillaries have these two ends. Over here the same thing, that these are all capillaries. Although they do not have an arterial and venous end, these are unique capillaries. They have arterial end and arterial end, both are arterial end. Anyways, important thing is, under this pressure the flow of substances occur from these capillaries into the Bowman space. That is what is happening. The whole thing's primary function is to do this, is to form ultrafiltrate. So write it down somewhere, ultrafiltrate. What is the definition of ultrafiltrate? Ultrafiltrate is really plasma being filtered under pressure, and in case of renal system, ultrafiltrate, the substance which comes here, this substance is actually the same as plasma, except that it does not have proteins in it and it does not have lipid bound substances in it, for example bilirubin and others. So once again, ultrafiltrate is nothing but it is the same thing as plasma. So the concentration of substances are the same as plasma, except it does not have proteins in it. Proteins stay here, lipid substances stay here, or lipid bound substances, for example bilirubin, for example T4, these stay here. But other substances, sodium comes in here, chloride comes in here, hydrogen comes in here, bicarbonate comes in here, small amino acids come in here, glucose arrives here, water comes in here, urea is filtered here. So that all is ultrafiltrate. Now this ultrafiltrate is exuding, the things are coming out of the blood system